Hello student. Today we are discuss about genetic code and its characteristics. This is the topic included in the BSc part first, Zoology paper second, cell biology. What is the genetic code and characters of its? Myself Dr. Neeraj Saini, Associate Professor, Rajasi Government PG College, Alwar. First of all, we discuss about flow of genetic information. You know, DNA have a process that is replication, and after that transcription, mRNA formed from DNA, and mRNA translated into protein. This is the flow of genetic information. The central dogma of molecular biology describes the two-step process, transcription and translation, by which the information in gene flow into proteins. So DNA into RNA, RNA into protein. This is the central dogma of molecular biology, or flow of genetic information from DNA to protein. This is a type of mRNA, 5 days and 3 days, and tRNA. This is the polypeptide chain, peptides, polypeptide chain. The pathway of protein synthesis is called translation. Because the language of nucleotide sequence on mRNA is translated into the language of amino acid sequence. There is a nucleotide in mRNA which are translated into amino acid, which form peptide form and formed a polypeptide chain. The process of translation requires a genetic code. This is the process of translation require a genetic code through which the information contained in nucleic acid sequence is expressed to produce a specific sequence of amino acid. What is the genetic code? The letter A for adenine, G for guanine, T for thymine, C for cytosine correspond to the nucleotides found in DNA. They are organized into codons. The collection of codons is called genetic code. What is the genetic code? The collection of code is called genetic code, or codon is A, G, T, and C. For 20 amino acids, this 20 amino acid is responsible for protein synthesis. For 20 amino acid, there should be 20 codons. Each codon should have 3 nucleotides to impart specificity to each of the amino acid for a specific codon. This is the double helix structure of DNA, and this is the one strand or 3 bases code for one amino acid. 3 base ATG 1 codon, CTA 1 codon, GGC 1 codon. There are 3 types of amino acid. If we decide the one nucleotide for a codon, only 4 combination is present. If 2 nucleotides, there is 16 combination. If 3 nucleotides for a codon, 64 combination are present. This is the most suited for 20 amino acid. So genetic code is dictionary that correspond with the sequence of nucleotides and sequence of amino acid. Words in dictionary are in the form of codon. Each codon is triplet of nucleotides, 64 codon in total, and 3 out of these are nonsense codon, or 61 codons for 20 amino acid. These 61 codons is sense codon and 3 are nonsense codon. Okay. There is a table of genetic code. This is the first codon, GACU or UCAG. There is UCAG UCAG or UCAG. Third codon, 3 letter. Yeah. The combination of these 3 letter, there are 1664 codon, right? In which AUG is initiation codon for methionine, or this is the UAA, UAG, or UGA is the stop codon. Stop codon. Genetic code is characters. Specificity. Genetic code is specific, non unambiguous. Not ambiguous. This is the unambiguous. So genetic code is specific for specific letter for specific codon. A specific codon always codes for the same amino acid. Always same amino acid. For example, UUU codes for phenylalanine. It cannot code for any other amino acid. This is the specificity. Universal. In all living organisms, genetic code is the same. The exception to universality is found in mitochondrial codon, where AUA codes for methionine and UGA for tryptophan, instead of isoleucine and termination codon, respectively, of cytoplasmic protein synthesizing machinery. This is the only a exception. Otherwise, genetic code is universal. AGA and AGG code for arginine in cytoplasm, but in mitochondria they are termination code. These are exceptions. Otherwise, genetic code is universal. Genetic code redundant. Genetic code is redundant, also called degenerate. Although each codon corresponds to a single amino acid, but a single amino acid can have multiple codons, except tryptophan and methionine. Each amino acid has multiple codons. A single amino acid, but a single amino acid can have multiple codons. Each codon for a single amino acid, but a single amino acid is have a multiple. There is the UUU, UUC 2 codon for phenyl, leucine 4 codon, methionine only single. There is 4 codon for one amino acid. So there is a single amino acid can have multiple codon. Genetic code is non-overlapping and non-punctuated. All codons are independent sets of 3 bases. There is no overlapping. कोई overlapping नहीं होती है. Codon is read from a fixed starting point as a continuous sequence of bases taken 3 at a time. The starting point is extremely important, and this is called the reading frame. Nonsense question. There are 3 codons out of 64 in genetic code which do not encode for any amino acid. These are called termination codon or stop codon or nonsense codon. The stop codons are UAA, UAG and UGA. They encode no amino acid. The ribosome pauses and fall off the mRNA. This is the stop codon, termination codon. Ochre, umber and opal. Ochre, opal, ochre, umber or opal. Initiator codon. AUG is the initiator codon in majority of proteins. In a few cases, GUG may be the initiator codon. Methionine is the only amino acid specified by just one codon, AUG. Bubbling phenomena. The rules of base pairing are relaxed at the third position. There are relaxation, so that a base can pair with more than one complementary bases. Some tRNA anticodon have inosine at the third position. Inosine can pair with U, C or A. This means that we don't need 61 different tRNA molecules. Only half as many are required. First two bases in codon in mRNA 5 days to 3 days base pair traditionally with the second and third base of the anticodon in tRNA. Non-traditional base pairing is observed between the third base of the codon and first base of the anticodon. The reduced specificity between the third base of the codon and the complementary nucleotide in the anticodon is responsible for bubbling. tRNA, that is an example for explaining the bubbling phenomena. tRNA first base, this is the tRNA first base. mRNA third base, traditional CG traditional, AU traditional, UA traditional, UG non-traditional, GU non-traditional, IU non-traditional, that is a non-traditional. That is a base pair. Third position पे changes is a third position changes is called the bubbling phenomena. Protein has 4 codons. We can explain. Or protein has 4 codons. That is CCU, CCC, CCA, CCG. There is a first and second codon similar, C and C, but third base pair is different. This is the bubbling phenomena. But CCU, CCC, CCA, CCG is 4 codon for protein. The first 3 codon can be recognized by a single tRNA having inosine at the first place. There is some clinical significance. Mutation can be well explained using the genetic code. There is 2 types of mutation: point mutation, frameshift mutation. Point mutation is silent mutation, missense mutations, or nonsense mutations. Silent mutation, single nucleotide change A to G, same amino acid is incorporated, mutation goes unnoticed. Silent mutation, there is only one third base जो है change A to G, then A to G, glycine, that is a unnoticed silent mutation. Missense mutation, single nucleotide change A to C, different amino acid incorporated, loss of functional capacity of protein. There is amino acid, a type of histidine, histidine amino acid, replacement of the single nucleotide, that is a single nucleotide A to C, there is proline, instead of histidine. Incorrect amino acid which may produce a malfunctioning protein. This is the missense mutation. Nonsense mutation, single nucleotide change from C to T. Stop codon is generated in mRNA represented by UAG. Premature termination of chain may be incompatible with life. There is a amino acid chain. Replacement of the single nucleotide A to T. There is a TAG is stop. Incorrect sequence cause sorting of protein. There is a stop codon. AG, stop codon. There is nonsense codon. Frameshift mutation, insertion or removal of a base can alter the reading frame with the resultant incorporation of different amino acids. G, frameshift mutation, G, AAG. This is the G replaced there. AAG, AAG, CAG, CAC, and GT, only GT. This is the frameshift mutation. Summary: genetic cause is unambiguous, universal, degenerate, comaless, and non-overlapping. UAG, UGN, UAR termination codon. AUG is the start codon. Mutation can be well described using genetic code. Okay. Okay. Thank you.